I'm Anna Dumitriu, and I'm going to talk today about engaging people in science and scientific topics through bioart. Bioart is a kind of art form that uses living materials or biological substances in its manufacture somehow. How did I get into this? I've been fascinated by science for a long time and the scientific process, but I trained as an artist because that's how I understand the world. I see art as a kind of meta discipline that can bring together lots of different disciplines from history, philosophy, ethics, and science all together, as well as the aesthetic sensations that we feel and the ways that we respond to the world. This is a fairly early example of an experiment that I did to see whether we could grow MRSA bacteria on calico, and if we sterilized it, would it look the same? And so we use these, it's called a brilliance agar, which only grows blue in the presence of this MRSA, or as it's called in America, MRSA bacteria, which is a Staphylococcus aureus that's drug resistant. And it worked. And I was able to make a quilt, for instance, like this. This is my antibiotic resistance quilt, which is impregnated with the World Health Organization's top 10 superbugs. And the actual color of them is the bacteria growing on these different chromogenic agars, and then it's all hand stitched together. But they've been sterilized, so the bacteria was in the making, but there's no live bacteria left. But there's still this sensation that you get when you approach it. And that was originally commissioned by the Science Gallery in Dublin. Other projects I've done, I was fascinated in this issue of antibiotic resistance, the idea that the bacteria that we use, we treat with these antibiotics, and we don't have these problematic infections like they used to do in the olden days when you'd cut yourself and you might die from that. They're stopping working because they're evolving resistance due to a lot of use of antibiotics, particularly in the agriculture industry, in things like farming, in factory farming and things like that. And so I was interested in, could we use new technologies to knock out or remove antibiotic resistance from bacteria? And so I did an experiment about that. And I worked with the Museum of History of Science in Oxford and Technion in Haifa to create this project, and I was inspired by their collection at the Museum of History of Science in Oxford, where they have the fermentation bottles and things like that that Florian Chain used to create penicillin, the first penicillin drug. As we all know, Alexander Fleming spotted penicillin and realized it might be useful, but he actually abandoned the research in 1929, saying it was too difficult. But as the war was coming and things like that, Florian Chain thought it was really important that we would have something to treat these diseases, particularly gonorrhea, incidentally, not the wounds, battlefield wounds that we think that they were mainly working on, but the UK government needed it to treat sexually transmitted diseases, which is an interesting perspective. And around 1941, when they first got penicillin working in a human, the UK government printed this leaflet, Make, Do and Mend, and it said that you should make patches on things and unpick things and knit them again, and if there was a moth hole in it, you should repair it. And around that time, I was also working with Modernising Medical Microbiology in Oxford, and this piece, it's made in the same way as the one that you saw previously with the Petri dish with the bacteria growing in it. But these are actual fecal samples from patients in Oxford that have been treated with antibiotics. And so they have a depleted gut microbiome. This is called a UTI agar, urinary tract infection agar, and it grows pink for E. coli, blue for Enterococcus, and you get small and big colonies and things like that. So here you can see these different kind of responses to antibiotics that different people have. Some have just E. coli, some have just enterococcus, some are a nice mixture still, so they're still quite healthy. And then I've embroidered it on there. It's been sterilized. I've embroidered onto it the images of E. coli bacteria, and these are ampicillin susceptibility discs, which they use to test whether bacteria are affected by antibiotics. And this actually made it onto the cover of the Centers for Disease Control Emerging Infectious Diseases journal. So so these art projects can have a big impact in the science world. And I was fascinated, could I actually remove this antibiotic resistance gene and repair it somehow? So I learned about CRISPR DNA editing back in earlier than about 2014, 2013. But in 2015, I was able to go to Haifa and work in Technion University there. And I was able to work with them to learn how to edit out the ampicillin resistance gene on an E. coli bacteria, which is a derivative of penicillin. And I repaired it. I wrote in DNA and made a repair fragment for the DNA. I wrote the phrase make, do, and mend. So it has this metaphor of patching that that was in the leaflet. So could we repair it? And the interesting thing is, around that time, so this is the first time we got it working, and we knew we had the make, do, and mend strain, so we put it E. coli M&M it's called there. I had 4 different vials of it there, so it was a very exciting moment. And the UK government launched in also 1941, they launched a branding that they stamped on furniture, clothing, and anything that was considered sustainable during the rationing period. So this suit here is one of these things. So it was stamped with this, and it was CE41, CC41, controlled commodity, 1941. It meant it was a controlled commodity. But of course, penicillin wasn't a controlled commodity. Antibiotics haven't been a controlled commodity. 70% of the antibiotics used in the world are used in agriculture, in factory farming, and we need to be much more careful about how we can use antibiotics sustainably, so we don't drive evolution and drive these bacteria to become resistant to it. Because if we don't have working antibiotics, then things like transplants, cancer treatments, things like that will not be able to work. So it's a really important issue. It's a globally important issue. So this piece was all around that. You can see the leaflets there, you can see the suit, and I grew the top 10 E. coli onto the material and patched the holes in this 1941 suit, and I was able to show the repair fragments and the antibiotics. So this vial here, this little bottle, that's 1 disc for every antibiotic that Oxford Hospital currently has available to treat diseases. So it's not a huge antibiotic arsenal that we think. These are the ones that they use. And the other bell jar that you can see just behind had in it the CRISPR repair fragment and the molecular scissors in it, dried into paper. Because although in the UK and in the EU you're not allowed to show genetically modified living organisms very easily, you can, but it's complicated, very complicated, prohibitively complicated in many ways. You can show the repair fragments, you can show DNA isn't covered in these same ways. So so that was the installation, and this was shown at Beaux-Arts in Brussels, and it's been shown at the CDC Museum in Atlanta, Georgia, and Oxford Museum of History of Science, among others. I'm the artist in residence with something called the National Collection of Type Cultures, which is the oldest collection of pathogenic bacteria in the world. And this is me at Porton Down, which is the most secure laboratory in the UK, holding a Petri dish in the Category 3 lab. And the Petri dish contains Staphylococcus aureus, which we've mentioned, and Yersinia pestis, which is the bacterium that causes plague. So this is, I was able to go there. I was invited there as an artist working deeply in this field to learn about how they work with what they call potential deliberate release category 3 agents. And I have a certificate now in handling these agents to say that I've done it and that I know about it. So it gave me a great experience of learning how to work in these sealed cases under negative pressure, and work with these very hazardous substances. Obviously, I've worked a lot with category 1 bacteria before that, and category 2, which are slightly less dangerous. And these this is the highest level of risk for bacteria. There's category 4, but that's only viruses. So this is me handling it, and it led me to make this, which is the plague dress, and it's a 1665 dress design dyed with walnut husks, which Culpeper the herbalist said was a good treatment in the Great Plague era, or the Great Plague, actually. And it's dyed with walnut, and the embroidery on the front is an actual period embroidery from the era, and I've impregnated that with the extracted DNA of Yersinia pestis plague, which I extracted from killed plague at Colindale in London at the National Collection of Type Cultures. So it's the oldest collection of pathogenic bacteria in the world. And then the dress is stuffed full of lavender and turmeric and other spices, and lavender all over the floor. So when you go and see it, it's a very sensory experience, because you're getting the smell as well of the thing, but there's the frisson of this actual embroidery that was around at the time of the Great Plague, impregnated with its DNA. And there's a close-up of the embroidery. And that's it on show at the Boerhaave Museum in Leiden, which is a perfect location for it, because it was actually a pest house, a plague hospital, in its previous instantiation. So that was on show for like 2 years. It opened July 2020. It was actually an exhibition that was supposed to be about the coming disease X, but unfortunately the disease X, which was, we know, of COVID-19, came slightly before it. So the opening of the exhibition was postponed, and it was opened by the King of the Netherlands in July 2020, and it was on show for a couple of years. So that was very nice, that even though there were the closures, people could go and see it and think about this, and it was a lovely part of the show. As I mentioned, I'm the artist in residence at the National Collection of Type Cultures, and they store bacteria. They've been going for over 100 years, and they store bacteria in a freeze-dried state. So what they do is they culture the bacteria, they whole genome sequence it, and then it's stored in these vials, and people can actually buy it. So a Staph aureus that I worked on, that's from my actual body, and I've whole genome sequenced and everything like that, is now in the National Collection of Type Cultures. So in perpetuity, they will keep it alive in perpetuity in this freeze-dried state. And it's the first bacteria that they've ever collected for its artistic importance, and that's mentioned on their website as well. So it's a kind of way of an artist intervening in the science as well, and art being very present within the science, which I love. So if there are any bioartists of the future out there who want to work, it's a bit like working with the paint of a previous artist, but you can work with the Staph aureus from this bioartist. This is a work I made during the COVID-19 pandemic. It's called Shielding, and it was commissioned by the University of Brighton, and it references the temporary hospitals, but also the issue of women being shut in, or people being shut in with abusive partners, which was a kind of hidden pandemic during the pandemic, when people weren't able to go out or get away from other people. So it's sort of telling this story, and I live very near where Virginia Woolf lived, and she wrote a famous essay called A Room of One's Own. And she said to be a writer, you needed an income of £5 a year and a room of one's own with a lock on the door. And so I was referencing that issue in it, and the tiny beds, which are hand embroidered and hand dyed with natural antimicrobial dyes, are all impregnated with the extracted DNA of SARS-CoV-2 from a plasmid construct. So it's not the infectious agent, it's actually a reagent. So it's just the DNA, but it is there. So I love this idea of the presence of it, that even in a safe way being part of the artwork. And I was able to do an Institute of Advanced Studies fellowship at the University of Surrey, and I was able to work with Christine Rollier there, who has created a plague vaccine using the Oxford University platform that is called the ChAdOx1 platform, which is what they made the Oxford AstraZeneca COVID-19 vaccine on. It's an adenovirus platform. And this piece is about the sort of history of the plague and the treatment of the plague. Paracelsus, the ancient physician or the medieval physician, said that he thought that plague was such an issue that instead of having a kind of broad spectrum, as you might call it, an antibiotic, a broad spectrum amulet, because this was their arsenal to deal with diseases, was to wear an amulet, that you would make a special plague amulet that was purely targeted to warding off the plague. And so he said that it should probably contain toad. So you can see my amulet looks like slightly like the back of a toad and the head of a toad. He said it should probably contain sapphires. So I used lab-grown sapphires here, or amber if you couldn't afford sapphires, but lab-grown sapphires are available these days. He said it should contain menstrual blood. So we have the drips there. And I used a ribbon that was dyed with the walnut, as same as the plague dress. But it's really interesting that the development of science and how it goes, because after Paracelsus, he said, I won't come up with it in my lifetime. I don't think I'll come up with it. But future scientists or future natural philosophers will come up with it. And they progressed the science, and they said, well, not a whole toad, that's not going to work. You want to powder the toad. So powdered toad. Menstrual blood is fine, but it should be the menstrual blood of a virgin. And then someone else said the menstrual blood of a virgin gathered at a full moon. So we have a full moon on the amulet as well that you can just see. And it should be gold, of course. So this is the amulet that now contains the ChAdOx1 platform plague vaccine. So hopefully we're kind of saying that Christine has actually come up with an amulet that will ward off plague, and this is the kind of the ultimate end of that journey. There's another image of it close up, and you can see that the amulet has a space inside, and the plague vaccine is inside it. So you can wear it and you have the plague vaccine. And this is shown at the Nobel Prize Museum. Well, actually, it was shown at Liljevalchs in Stockholm in an exhibition called Life Eternal that was curated by the Nobel Prize Museum. I've worked with scientists to explore ancient DNA, and this is, I actually learned hands-on how to extract ancient DNA from Siberian permafrost human remains, Iron Age human remains, over a 3-day period, working hands-on with the scientist. And I learned how they cut squares from the bone. And this bone is decorated. It's a laboratory prepared bovine femur that I've carved and painted with the images of the bovine TB. And then I used the ancient DNA that I extracted from the human remains, because we did find it in the sample, and impregnated the bone with that. And you can see close-ups of the details there. I work with lots of wet, smelly stuff sometimes. And this is a bit of the gut model at Leeds University. They have a whole room full of gut models that they seed with fecal samples and then add Clostridium difficile bacteria to, which is another superbug that causes bad diarrhea. And then they give it antibiotics and see what happens and study the process. And I wondered if we could grow linen, grow the biofilms of the C. diff, which are the causative agent of the disease. I wondered if we could grow those onto crocheted linen. The reason I used linen was that the pioneering microbiologist Sergei Vinogradsky, who looked at these Vinogradsky columns, they named after him anyway, but he made these bacterial ecosystems which are very much like the gut, only in the environment. And I highly recommend people listening to this, look it up and go and make their own Vinogradsky columns, because they're fantastic. And here we grew the Clostridium difficile biofilms on it, and we were able to autoclave them on a kill cycle that destroys the spores. And I wove them into this woven linen collar, and here they are, and these are the biofilms, and they're quite hard. It's quite an interesting work, and I made other works that reference Vinogradsky. He discovered the first nitrogen fixing bacterium, and he discovered the bacteria that are responsible for the retting of flax in linen production. And so this is made with flax, and it's made with the bacteria and some human hair, referencing the idea of flaxen haired, these hair wreaths that are an antique craft, and also flax seeds in there. And that piece is called Pastorianum, which was what he called the bacterium first of all, Clostridium pastorianum. Then they changed the name later to Pasteurinum after Pasteur. But Pastorianum means from the field. So I love this idea that it's a bacterium from the field. And Clostridium means spindle, so spindle from the field. I love looking into these kind of background stories and then weaving them into the artwork. And I find it really deeply engages the audiences. More works with, this is called Bacterial Baptism, and it's about how when we're born, we get a flood of bacteria, and we swallow the bacteria from our mothers, so it's a christening gown, but it's also impregnated with the DNA and with different bacteria, different embroidery silks dyed with bacteria, dyed with laboratory dyes actually, and the spindle like crochet with linen is also on there. And this has been shown in numerous galleries now. I always work hands-on. It's really important to me. Though I trained in fine art, I think it's really important to always work hands-on in the lab. So I learn all the processes because I think it helps me to communicate it. And this is me at Boku University, in the Institute for Microbial Biotechnology, learning how to modify yeast, and working with Professor Ditard Matanovic there in Vienna. We created this project, myself and Alex May, called Fermenting Futures. And this is kind of a vessel based on a fermenter that they use in the lab, like the one I just showed. But the yeast that's in it is able to capture carbon from the environment and output lactic acid, which can be polymerized to make polylactic acid, or PLA, which is 3D printing filament. So it's like you can capture carbon and you can output plastic. And this little bit you can see in the installation, there's quite a lot of 3D printing and stuff in there, but it's actually this one here, the white kind of blob there, that contains the lactic acid that was made by the yeast. So it needs improving, obviously, but it is kind of working, and the scientists are working on lots of different kinds of biotechnological solutions for environmental issues that allow us to produce useful materials such as nylon sustainably. And we show it's on show at ZKM in 2023, and they've done a display where they've shown the yeasts and some of the things made from the PLA that the lab has made, and a display of different things in the exhibition. So it combines the art, but also images of the science. So this is an interesting way of working with the museum. We worked with them to discuss what they should contain in the show, what it's needed to create this dialogue between the art and the science, and visitors can learn about the science, but also reflect on the issues around this. I've been working with the Helmholtz Zentrum in Munich as well, and I made this piece about epigenetics, and we've been learning about stem cells and how stem cells can be differentiated, how cells know what kind of cell they're going to be, because you get an original cell as a kind of totipotent cell, then it can be anything. And so what does it decide to be? How does it know it needs to be, bit of your head up here? How does it know to go there? And can you reverse that? These are the questions that they're working with. So there's one gene called OCT4, which is known as a Yanamaka factor. So it's important for making stem cells. And I made a necklace from this OCT4 gene. So it's the exact scientific representation of it in the form of a necklace. So we've got these histones that are wrapped with the DNA, which is the thread, and the ribbons that are tied together are called the, for scientists in the audience, H3K9 trimethylation, and they're kind of connected all there, but it's anatomically correct model, but beautiful. And when I was working in the lab, they said that I needed to use copper sulfate sometimes because cells needed copper sulfate to grow. So the necklace was then dipped in copper sulfate. It's very fiddly to make, incidentally, because making a necklace that does what DNA does is very, very tricky, because DNA is, you know, has got these kind of, there's sort of magnetic forces between the histones and the DNA naturally wraps around the histones, and you can't do that by hand. There's a kind of detail of the piece, and so what I did after I made the first one, which was kind of hell, I had a few very fiddly work. So I had a few other exhibitions that wanted it simultaneously. So I came up with the idea of 3D printing it again and getting all the scientists together in the laboratory, in the breakout room, and we did a workshop where we made the necklaces together. So they learned and I learned as we were doing the process, and it was able to go off to more exhibitions. So then I got invited to do a project about chicory, about genetic modification of plants, and is the EU ready for it? So it was part of an EU project called CHIC, and I went to work with them, and we were reflecting on the idea that Goethe was very inspired by the chicory plant. In German Romantic movement, there's a book called Heinrich von Offerdingen by an author called Novalis, who he didn't finish the book. He died of tuberculosis before he finished the book at age 20. It's a very romantic book, and in it he talks about women crying on the roadside and chicory plants bursting forth in bloom from their tears, and he talks about well, actually, that in other things, in witchcraft mythology, the chicory plant isn't described as the key that opens locked doors. So I thought this is really interesting in the sense of are we allowed to, should we be allowed to do what they now call precision breeding? In the UK, they've just passed a law, a precision breeding act, to make it more straightforward to do it. And in the EU, they're allowing it for animal products, animal feed and things like that, because in a situation of climate change with different pests and things like that, it's possibly a sensible way to go. You can think about that and you can debate that amongst yourselves. But I learned hands-on how to modify chicory. I should say that Goethe wrote Metamorphosis of Plants, and he was inspired by his interest in plants was inspired by Novalis's writings, and that led on to Darwin disagreeing, but taking that next step. And Goethe said that the plant comes from you grow the plant from the leaf, just the leaf. But actually, I learned that you can grow the plant just from the cell. And I learned how to CRISPR modify. And these are CRISPR modified cells. And we knocked out some of the bitter terpenes in the plant protoplast. So we can grow chicory plants that have less bitter flavor. So this is wild chicory, not the lettuce type chicory. So they can be used to produce inulin more cheaply, and inulin's a very good dietary fiber and sweetener. And it was shown at Rome Botanical Garden, but also I saw a open call on the internet saying that MIT Museum was relaunching, and they're building a whole new museum, and it's relaunching with a Bioart exhibition, and there was an open call for artworks. So on gene editing, so I submitted the chicory project, and it's on there until 2024 for several years. So so the artwork has this fantastic outcome with thousands and thousands of visitors coming to this major museum in Cambridge, Massachusetts, near Boston, which is it's a very major, new and flashy museum. So that was fantastic. And there's another image of the show with the chicory plant that we made, which is a 3D printed chicory plant. I worked with Alex May and worked with the EU Chick project, and it's about this idea of digitization and Goethe said this phrase, the unnatural that too is natural, which I love. And the center of the flower in the chicory plant it has the chicory protoplast in it, and behind it we edited a genome of the chicory plant and removed all the repeating sequences, because Goethe said that that he believed that there was an original plant from which all other plants evolved, and he called that the Upflanze. And it's and this is the Upflanze genome from our perspective. And I would love one day to actually make that genome in DNA. There's also an app, the Blue Flower app, which you can find on the Google Play Store, Blue Flower, or the Apple App Store. And you can plant genetically modified chicory plants digitally wherever you go. I've just completed a project on fava beans, and it's on show now. And these are fava beans, and they're the weirdest. They've got the weirdest story behind them. So that past project with the plants led on to this. And there's something called the protein transition. So it's the idea that we're eating too much meat, and we need to eat a bit less meat and a bit more vegetables to get our protein from. So at the moment we get 60% of our protein from meat, but they want to make it so we get 60% from vegetables. And fava beans are a fantastic source of protein, but they have some problems. 4% of the world's population have a gene that gives them a terrible form of anemia called favism if they eat fava beans, or even if they smell them sometimes, if they've got a severe case of it. So that's a big problem. Also, it has a protein uptake inhibitor gene in it. So it actually, the protein in the fava bean isn't bioavailable to us to actually use to make flesh and stuff like that from. So I worked with scientists at Keygene again, where they were working on it, and I learned all about it. And this is the show that literally opened a couple of days ago, and it's in a location called Artfy in the Netherlands, in quite a farming district. And this is a series of works that I made that have different stories about the fava beans. There's a lucky bean necklace that contains the DNA of 4,000 pooled plants, flowers. One of them is from a plant that they've managed to create that doesn't have the gene that causes favism in humans and things like that. Victorians believe beans were lucky, so that's why it's called the lucky bean necklace. To get rid of the protein uptake inhibitor, you need to boil the beans, and this is actually a moving artwork, and the saucepan lids are boiling, so it's constantly bubbling, and the flames are constantly flickering in this toy oven that I've altered. The DNA of 4,000 plants, they work with 80,000 plants and pull the DNA and use a very interesting key point technology, they call it, to find the exact plant that has the mutation they're looking for. So it's not a CRISPR thing. They choose to do a traditional chemical mutation instead of CRISPR because of the paperwork. And there's a video. And talking about working with galleries and things, first of all, we wanted to do a projection. We couldn't do a projection because it was too light, so they bought a TV. But and they bought me a fantastic 80-inch TV, which was beautiful, but it was too heavy to go on the wall that they had it turned out when. But then we came up with this beautiful solution of covering the base. They was just fixing it to and putting the TV screen on the floor and covering the base with soil, so it looks like the projection of the video of the beans and things like that growing, and the research is coming from the earth, which was lovely. And then just to conclude, my latest project I'm working on is called Mysteries and Mycotoxins, and I'm working with Dr. Carol Vihicki-Varson at the University of Cranfield, and I got funding from the Massey Award of the British Mycological Society. So that's fungi and molds and things like that. And I'm working with molds that produce toxins, highly dangerous toxins that can be very deadly, carcinogenic, all sorts of things. And the most famous one is ergot, which you might have heard of. It's famous throughout history. In ancient Greece, they used it in a drink called kykeon at the Eleusinian Mysteries, where they had these fantasies and they got close to God, as they believed, or the gods in those days. And it's an amazing site that I visited. And they also believe that the Salem witches ate ergot and this in their rye bread because it grows on these cereal crops. They believe they ate it in rye bread, and it led to the behaviors that led to the witch trials and things like that. And this is a picture of some ergot that I actually collected with a scientist safely from a field in the UK, because damp weather means it's present quite a lot around. So we're going into a deep exploration of this. And the way I'm doing it, because it's highly dangerous, you can't kill it through autoclaving like I do with my bacteria, but you can kill it through putting it in a furnace and turning it to ash. So I'm working with the ash from this to make paintings, and that's basically what I'm going to be doing for the next few months, and working with different materials from this. And that's just a kind of whistle-stop tour through my artwork, and I hope you've enjoyed it. You can see more on my website, www.annadumitriu.co.uk, and there's some links to my work and some of the supporters. Thank you.